This animation is brought to you by Lab Group 4 with members Catherine, Kira, Nicole, and Ian. We're going to teach you about cellular respiration, specifically the last step, oxidative phosphorylation.
Oxidative phosphorylation uses NADH, FADH2, and O2 to produce ATP energy in the electron transport chain and ATP synthase. The ETC is located in the inner membrane or cristae of the mitochondria. Oxidative phosphorylation occurs in aerobic conditions, meaning oxygen is present. This reaction creates about 32 to 34 ATP molecules per 1 glucose, largely contributing to the overall amount of ATP produced per 1 glucose molecule in cellular respiration, which is about 36 to 38.
The inputs of oxidative phosphorylation are 32 to 34 ADP, 6 oxygen molecules, 2 FADH2 molecules, and 10 NADH molecules. The outputs of oxidative phosphorylation are 6 H2O molecules, 32 to 34 ATP, 2 FAD, and 10 NAD plus molecules.
NADH is oxidized to NAD+ and deposits its electrons in the first protein. These high-energy electrons from NADH are excited and jump from protein to protein until they have expelled most of their energy and become low-energy electrons. FADH2 is attracted to the second protein, therefore depositing its electrons there. These electrons become excited and turn into high-energy electrons which jump from protein to protein, expelling energy.
The low-energy electrons combine with nearby H+ ions and oxygen, the final electron acceptor, and oxidize O2 to form water, a byproduct of cellular respiration.
Step 2. When NADH and FADH2 are oxidized in the mitochondrial matrix, they release their hydrogen ions. Then, using the energy given off by the high-energy electrons, the H+ ions perform active transport, which is the movement of ions from an area of low concentration to an area of high concentration by using added energy. The ions travel from the mitochondrial matrix, which has a low hydrogen ion concentration, through the enzymatic transport proteins to travel to the inner membrane space, which has a high hydrogen ion concentration. This process of moving hydrogen ions through active transport creates a concentration gradient, which is later used to power ATP synthase.
The concentration gradient is used to passively transport H+ ions through ATP synthase during the process of chemiosmosis. Chemiosmosis is the flow of H+ ions across a membrane from an area of high to low concentration to form ATP with assistance from the ATP synthase complex. ATP synthase complex helps phosphorylate ADP into ATP, allowing H+ ions to flow through the membrane, and uses energy released from the electrons moving down the ETC to power the complex.
The process fundamentally is O2, NADH, ADP, and FADH2 being used to make ATP with a waste product of H2O. It does so by creating a high to low H+ concentration gradient, where electrons are shuttled through the electron transport chain to release energy and eventually form ATP using the enzyme ATP synthase.